Hello, my name is Trina Ganguly, and the date today is January 31st, 2023. So today for my presentation on the prokaryotic and eukaryotic interactions, I will be discussing how bacteriophages engage in direct interactions with eukaryotes through horizontal gene transfer, or also known as HGT. So to begin with, I want to give some background information first. HGT is defined as a transfer of genes from one organism to another unrelated organism. And this transfer can occur between organisms from the same species, different species, and even organisms representing different domains. HGT occurs primarily in bacteria through three pathways, which are transduction, transformation, and conjugation. And HGT is common between bacteriophage and the bacterial host, and also between bacteria and eukaryotes, particularly among the obligate intracellular bacteria and their respective eukaryote host. And so in this slide, I'm going to be getting into the specific example a little bit and explaining that before I show the image. So here I am talking about the nuclear localization signal, which is a specific sequence that allows an uptake and delivery of proteins into the eukaryotic nucleus. And thus these TpDNA molecules are a useful tool when it comes to amplifying and subsequently delivering genes efficiently into the eukaryotic nucleus. TP is a protein used by bacteriophages to prime the DNA for replication. And in that process, TP is covalently bound to the 5' end of the DNA product. And therefore, following TP-mediated DNA replication, the TP-bound bacteriophage DNA product has a nuclear localization signal and can then enter the nucleus. This mechanism enables direct HGT between bacteriophages and eukaryotes, and also enables eukaryotes to obtain bacteriophage genes. However, for this kind of HGT to occur, the bacteriophage has to penetrate eukaryotic cells before releasing the TP-bound DNA. And so as a result, bacteriophage penetration through transcytosis plays a vital role when it comes to allowing direct HGT to occur. And so in this image, we see a drawn model of TP-mediated HGT. Please excuse my drawing skills. And so in natural ecosystems, HGT within prokaryotic or archaea organisms is often mediated by phages that might contain or not contain TPs in their genome ends, as depicted within the areas of 1A and 1B on the left side of the image. And infected bacteria contain a large number of viral particles in genomes and which might encounter with eukaryotic cells, and eventually might be internalized by phagocytosis or other processes, as depicted in parts 2A and 2B. And normally, viral DNA would be degraded inside the cell, as shown with 3A, but if the phage DNA contains 5' linked TPs, it may go through lysosomes and endocytosis pathways, as shown with part 3B, and subsequently be driven across the nuclear envelope by the NLS of the TP, shown with part 4. And eventually be incorporated into cell genome, which is shown at the end in 5. And because phages mediate HGT between different bacteria, they may contribute to the incorporation of genetic information from themselves through the B parts or from the prokaryotic host shown through the A parts of the image to the eukaryotic genome. And here are my references. That is the end of my presentation. Thank you.